and we're going to screen sharing. And we're going to look at a new group of organisms, one that in some way we're a little more familiar with than probably the ones that we've talked about before this point, but as we'll see, one that in other ways, it turns out to have a lot of surprises. We have a lot of assumptions about animals, mostly that animals are things like people, and many of them are not. So depending on whose classification scheme that we want to follow, there are about 35 phyla of animals. Remember that the phylum is the subdivision under the level of a kingdom. There are a few people who have more than 34, a few people who say there are less, but 34 is a pretty good round number. Even though there are 34 phyla of animals, if you would take a walk through, say, the woods in Illinois or through the prairie, or if you would go scuba diving in a coral reef, or if you would go up to the Arctic tundra or a tropical rainforest, almost all the animals you see would belong to 9 different phyla. And we're going to concentrate on those phyla. So here's a cladogram of animals. And notice down at the bottom, we have our common ancestor of animals, and the common ancestor of animals, as best we can figure out at this time, probably resembled those choanoflagellites that we looked at with the protists. Now, they were probably a colonial choanoflagellite, meaning that numerous of them lived together. Sometimes it's different. It's difficult to draw a line between organisms that are colonial and organisms that are truly multicellular. So the line isn't a real firm one, but usually when we talk about colonial organisms, they cooperate, but we don't necessarily have true tissues like we see in animals. Well, if we look at the 9 big groups of animals, the 9 major phyla, the first group to split off is Porifera. And as we'll see in a few moments, Porifera are basically just colonial choanoflagellates. They have a little more cooperation, so they're usually considered to be true animals, but some people actually don't classify sponges with animals. Now, once we look at the other groups of animals, and because the sponges are kind of off by themselves, they're sometimes put in a separate sub-kingdom. So some people talk about the sub-kingdom Parazoa. Zoa is animals, para, think of parallel lines. Parallel lines are next to each other. So the Parazoa are next to the animals. So some people classify sponges, the phylum Porifera, in a separate subkingdom. And then they put all the other animals into a second subkingdom, the Eumetazoa. And again, zoa is animals. Meta refers to the fact that these animals are multicellular, and eu means real or good. So good multicellular animals, as opposed to things that are kind of colonial. Sponges are kind of half and half colonial, half multicellular. So the first group that split off in terms of the main phyla is a group called the Cnidarians. And Cnidarians include things like jellyfish and coral and sea anemones. And what distinguishes them from the rest of the animals is that they have what we call radial symmetry. Their body is basically a circle, 3-dimensional, but probably radial symmetry would be better if it had been named diametrical symmetry, because the basic idea of radial symmetry is, since these organisms have a round body, if we cut a diameter through the body, it doesn't matter which direction we cut that diameter, we get 2 equal halves. That's not completely true, but it's mostly true. Now, the other groups of animals all have what's called bilateral symmetry. And bilateral symmetry, bilateral means there are two sides to the body, and the two sides are symmetrical. They're basically mirror images of each other. Keep in mind, like with radial symmetry, it's really only approximate. People are considered to have bilateral symmetry. But if you look at the right side of your body and the left side of your body, they're not exactly mirror images of each other. Most of you are probably familiar with the idea that your heart is slightly to the left side of your body. What you may not know is that your appendix and your gallbladder are slightly on the right side of your body. So the two halves aren't exactly mirror images, but pretty close. You have a right and a left ear, a right and a left eye, a right and a left arm, a right and a left leg. And these things are pretty much equal. Now you might be right-handed or left-handed, but that doesn't mean that you have more fingers on your right hand if you're right-handed. Now, if we look within the groups of animals that show bilateral symmetry, they're divided into two main categories. One is a group called the protostomes. And a little bit later, we'll look at how animals develop after the sperm fertilizes the egg. But when the sperm fertilizes the egg, that single-celled zygote starts to divides by mitosis, and eventually it forms a big ball of cells. And this ball of cells starts to get an indentation. And in the protostomes, proto means first, this opening becomes their mouth. Stome refers to the mouth. And then later they develop a second opening, which is the anus. In the deuterostomes, deutero means second. And in the deuterostomes, the first opening becomes the anus of the digestive system, and the second opening is the mouth. Notice that in the protostomes, the main groups we see are the platyhelminthes. Helminthes means worm, platy means flat, so these are flatworms. You may never have seen a picture, but I'm sure you've heard of something called a tapeworm. Rotifers you might not have heard about. We'll talk about them just very briefly. They're not one of the 9 major phyla, but they're listed on here because they're close relatives of the platyhelminthes. Then we have the mollusks, and the mollusks include things like snails and slugs and clams and oysters and mussels. And the mollusks include the largest animals that are invertebrates. In at the beginning of a word means not. Invisible is not visible. Invertebrates don't have vertebrae. They don't have a backbone. And the mollusks include the largest invertebrates, which are certain types of squid, which may get as big as 50 or 70 feet long. They also include octopods. Now, the protostomes also include the annelids. Annelids are segmented worms, worms whose body is separated into distinct divisions. A good example of this that you might have seen is an earthworm. All those little rings that you can see on the outside of an earthworm's body, those are segments. And depending on the species, once we get past about the first 30 segments or so, all the other segments are pretty much identical. There's also a group in here called the nematoda, and nematoda are roundworms. So notice we've said there are 9 major phyla of animals. 3 of those phyla are flatworms, the annelids are segmented worms, and the nematoda are roundworms. And actually, if we would look at all 34 phyla of animals, about 10 of them are different types of worms. So there's a lot of different ways to be a worm. And the other group, which is probably one of the 2 groups we'll spend the most time talking about, is the arthropoda. You probably know that arthritis is a disease of the joints. Arthropoda have jointed feet. Poda, like a podiatrist, refers to the feet. By extension, it also refers to the legs. So these organisms have jointed feet and legs. And this includes a lot of groups that you've heard of: centipedes, millipedes, arachnids, which includes spiders, but includes some other organisms as well. Crustaceans, lobsters, crabs. You might think those are mollusks, but if you think about it, they do have jointed legs. So they're arthropods. And it includes the one group that has more species than any other group of living organisms. The phylum Arthropoda includes the class Insecta, insects. Right now, there are about 3 million species that have been studied and classified. More than a third of those are insects. In fact, about a fourth of them are beetles, one particular type of insect. Now, the deuterostomes, there are only a few phylums in this group, and there are only 2 phylums that are very common. One is the Echinodermata. Echino means spiny, derma means skin, like a dermatologist. So these organisms have a spiny skin. The most familiar example of these is something called a sea star. A lot of people call them starfish, but they're not actually fish. But it also includes some organisms called sea cucumbers, sand dollars, sea lilies. Echinodermata at various times in the geological past have been very numerous. Most of them, not all of them, but most of them are benthic, which means they live on the bottom of the ocean floor. And there are still places where when you go scuba diving, you can see literally just fields of these organisms. Now, the other group that's a deuterostome is the chordata, the chordates. And chordates have a stiff rod along their back, something called a notochord. Now, the phylum Chordata is divided into several subphyla. And besides the Arthropoda, the other group that we'll talk about quite a bit is one subphylum of the Chordata. And this is a subphylum called the Vertebrata, the things that have vertebrae or backbones. And the vertebrates include 5 main groups. Includes fish, and it turns out there are lots of different types of fish. It includes amphibians, things like frogs, toads, salamanders. Reptiles, things like crocodilians, lizards, snakes, turtles, dinosaurs. It includes birds, which some people actually classify with dinosaurs because some dinosaurs evolved into birds. And it includes mammals. So fish, amphibians, reptiles, birds, and mammals. Those are the vertebrates. Everything else on this chart, the echinoderms, the arthropods, the annelids, the platyhelminthes, the sponges, the rotifers, the mollusks, the annelids, everything else is an invertebrate. And even the other subphylums of the Chordata are invertebrates. The only thing that contains vertebrates is one subphylum of one phylum out of 34 total phyla of animals. Happens to be the subphylum that we're classified in, so of course we're very interested in it, but we don't want to get the idea that most living things are vertebrates. Vertebrates turn out to be a successful group. There are vertebrates like humans that have very large populations, but the invertebrates are successful too. And some of them do things where they control their environment. Maybe not to the extent that people do, but we want to keep that in mind. Now, the most recent evidence we have suggests that animals originated about 700 million years ago. And they probably evolved from some organisms similar to those protist choanoflagellates that we talked about. But for about 140 or 150 million years, animals were mostly small things. They mostly had soft bodies, so similar to jellyfish or sponges or some of the worms. But about 560 million years ago, something called the Cambrian explosion occurred. And the Cambrian explosion is called an explosion because at this time, animals, especially large animals, underwent a very dramatic and a very rapid adaptive radiation. And all the phyla of animals that we see today evolved within a period from about probably 570 million years ago to about 540 million years ago. So if you go way back then, you can find ancestral vertebrates, ancestral representatives of the other groups. There were also a lot of other groups of animals that evolved that have gone extinct since then. Some people think when animals started to go through this adaptive radiation, it was kind of like nature was performing a big science experiment. And some of the animals, when we look at their fossils, the ones that are extinct now, they're really pretty bizarre things. But there were all sorts of animals evolving. Some of them weren't successful, so they went extinct. Now, the best evidence we have is that if we go back to the early history of the Earth, there wasn't any oxygen in the atmosphere. There was probably hydrogen. There was a lot of water vapor. There may have been some ammonia, some methane. And what happened was, as photosynthetic prokaryotes, cyanobacteria, sometimes called blue-green algae, as they evolved, remember that oxygen is one of the waste products of photosynthesis. So these photosynthetic organisms began releasing oxygen into the atmosphere, and it's probably somewhere around 560 million years ago that atmospheric oxygen reached current levels. And current levels are about 20.9%. So the air around you is about 78% nitrogen, and it's about 20.9%, almost 21% oxygen. So before this, lower oxygen levels probably prevented large multicellular organisms from evolving, just because for aerobic cell respiration, if oxygen levels are lower, oxygen doesn't diffuse from the air into your body as well. And then it doesn't diffuse from your blood into your lungs as well. So we think oxygen was probably crucial in this process. Now, all animals have a couple of characteristics in common. First of all, notice animals are still in the domain eukarya, so they have eukaryotic cells. All animals are motile, at least at some time during their life cycle. Remember when we talked about bacteria, motile means they can move. All animals are heterotrophic, mostly by ingestion, which means they eat their food. But there are some spiders that when they trap insects in their web, the spiders bite the flies and they inject their digestive enzymes. And then they liquefy that insect and they leave it in their web, and they come back later and they bite it again and they suck out all the nutrient broth, the soup that they've created. So that's really, you could think of it as ingestion, but it's pretty similar to absorption. All animals are capable of sexual reproduction. Now, keep in mind, there are a lot of animals that are also capable of asexual reproduction. And sometimes sexual reproduction doesn't occur all that frequently. Sometimes they mostly reproduce asexually, but all animals are at least capable of it. And all animals develop through an embryonic stage that we call the blastula. In people, it turns out that the blastula has some important uses. The blastula is formed after the sperm fertilizes the egg, and that single cell fertilized egg starts to divide by mitosis. One of the first stages, not the first, but one of the first stages it forms is called a blastula. And this blastula in people, not just people, other mammals as well, this blastula starts making hormones. And these hormones prepare the lining of the uterus in the mother so that it starts building a placenta. And so the blastula that makes these hormones are what we test for in early pregnancy tests. Because if the egg is not fertilized, then the egg doesn't develop into a blastula. So we don't have these hormones present. If the egg has been fertilized, we can test, we can detect these hormones, and we know that the egg was fertilized. So let's look at some of the different phyla of animals. And as with the sponges, and as with the fungi and the protists, and we're going to see lots of pictures because we would look at a few living animals, and we would look at some animals under the microscope, but a lot of what we would look at if we were doing this lab on campus would be preserved specimens. So we're going to see some photographs of living organisms. If we look at sponges, there are 4 classes of sponges. Sometimes people will say there are only 3 and they combine these 2 on the left into one class. Remember that the subdivisions of a phylum are called classes. So when I say classes, we're really talking about a technical biological term. Usually these 4 classes are all put in one phylum, the phylum Porifera, but sometimes they're divided into 2 distinct phyla because there really are some significant differences between them. And sometimes the phylum on the right is classified with animals, and sometimes the phylum on the left is actually classified with protists, and they're not considered to be animals. Remember how I keep emphasizing that, taxonomy, how we classify organisms, is very fluid and it's always changing. Right now, all of these are considered to be in the same phylum by most biologists, and that phylum is classified with animals. But if we go back in the past, at one time, sponges were classified as plants. Why? Well, they don't really move a lot during most of their lifetime. They're kind of anchored in place. And back when everything was considered to be either a plant or an animal, sponges seem to have more similarities with plants, particularly when we weren't able to look at cells through a microscope and couldn't see that sponges actually ate their food. Sponges are the first major group, the first phylum that diverged from all the others. So notice if we go back to our cladogram, sponges are less closely related to the animals than anything else is. Why? Because they separated the longest ago. They separated first. So if we look at humans, mammals are over here in the chordate group. Mammals are more closely related to flatworms than they are to sponges. Mammals are more closely related to jellyfish than they are to sponges. Mammals are more closely related to any of the other big groups of animals than they are to sponges. Sponges are the least closely related of all the animals. As I say, sometimes they're not even classified with animals. They're considered to be so different. Now, one of the ways that they're considered to be different is if we look at them in a lot of ways, sponges are colonial organisms rather than multicellular. And when we say they're colonial, one of the things we use as a kind of a basis to judge this on is their cells really have minimal specialization and they don't cooperate all that much. One way to think about this is colonial organisms. If you were thinking about people, colonial organisms is a bunch of people living in an apartment complex. where they kind of all live in the same area, but they may not interact all that much. People who, if we would think of people as being a multicellular organism, rather than living in an apartment complex, a multicellular organism would be a bunch of people living in the same household, an extended family where you might have 3 or 4 generations in the same household, where there's a much higher level of cooperation. Now, what do all the sponges have in common? Well, they all live in aquatic habitats. Some of them are marine, which means they live in seawater. Some are freshwater. If you would go out and go scuba diving in Lake Michigan or in Wolf Lake or in most relatively large lakes in Illinois, you could find some sponges. They are benthic. They live on the bottom of the ocean. And typically they would be attached to a board that had fallen in a lake, or a rock, or a branch from a tree, or something like that. When we look at sponges, they all have a body with either, our book calls it radial symmetry. Remember, we talked about the cnidarians a moment ago, things like jellyfish. So radial symmetry means they basically have a round body. Or a lot of people consider them to have asymmetry. Things that are asymmetrical don't have any symmetry. And the reason they kind of fit into both categories is if you could take a sponge and you could put it into water that was perfectly still, as the sponge cells divide and produce the sponge, they divide by mitosis. As they divide, they basically would produce a circular sponge. The problem is in nature, there are no areas in the ocean or in lakes where there's not a little bit of a current. And since there's a little bit of a current, the actual pattern that sponges take depends on the direction of the current. So their body generally ends up being asymmetrical. Now, all sponges get their food the same way. When we look at the adults, there's something called sessile filter feeders. Sessile means they're immobile. They stay in one place. Remember a moment ago I said, where would you look for sponges if you were scuba diving in Lake Michigan? You'd look for a board that had fallen to the bottom of a lake, or a tree branch, or a rock. So they attach to something and they stay in that location. And there are lots of living organisms that are filter feeders, and living organisms that are filter feeders all get their food the same way. They pump water through their bodies. Now, there are filter feeders that are chordates. There are filter feeders that are sponges. There are filter feeders that are worms. There are filter feeders that are mollusks. So the way they pump the water through their body differs, but what they always do is they catch small organisms in the water for food. You could think of it like some commercial fishermen fish with a net. They throw a net in the water and things get tangled up in the net. So let's look at the body of a sponge. So sponges are what's called diploblastic. Di means two. Notice that blastic comes from the same root word as that blastula, that developmental stage of the embryo that we talked about a few moments ago. So things that are diploblastic have 2 layers of cells in the embryo, and these 2 layers of cells give rise to the adult cells, the adult tissues. Now in sponges, these adult cells are separated by a layer called the mesohyl, sometimes also called the mesoglia or mesenchyme. And this material is mostly non-cellular. It contains protein fibers, mostly collagen, that are made by cells and then secreted out of the cells. It contains some things called spicules that are made by cells and again secreted out of the cells. And these spicules can be a couple of different types of material. They can be silicon dioxide, which is basically one of the main ingredients in glass. They can be calcium carbonate, Or these spicules can be protein, a protein called spongin, which is only found in sponges, of course. And then around that, there's a lot of polysaccharides. Remember, polysaccharides are large carbohydrates. So as a result, this mesohel layer basically has a consistency kind of like a thick ice cream or a jello type material. So let's see, I think I've got, yeah. Here's some pictures of the spicules in the various groups. One of the groups that makes silicon dioxide spicules, they have multiples of 6 points on them. So sometimes there are exactly 6 points. Sometimes there are 6 points, but they aren't equal size. Sometimes there are 12 or 18 points. So they don't look like perfect snowflakes typically. One of the other groups of sponges makes spicules made of silicon dioxide, but they're different types of shapes. They're not all six-sided. The sponges that make spicules of calcium carbonate, calcareous, those again are all different shapes. And the sponges that make spicules of spongin protein, the spongin protein forms a kind of a net. So we can look at this mesohel layer and we can see what type of sponges we're dealing with if we're not sure because of other things. Notice that sponges don't have what biologists consider to be true tissues. But on the other hand, if we look at the cells are more specialized than we usually see in colonial protists. So sponges are kind of halfway between a colonial organism and a true multicellular organism. So let's go back here for a minute and let's look. So we have a layer of cells on the outside of the sponge. And this layer of cells on the outside of the sponge forms a kind of epidermis. And the epidermis, when we're talking about the epidermis, is always the outside of a sponge. And these outside cells are called pinocytes. So this would be this kind of peachy colored layer here. The yellow you can see, let's magnify this a little bit. This yellow that you can see is that mesohel. There are some cells called porocytes that line holes leading into the inside of the sponge. These poles are sometimes called in-current canals, and this is how the water flows into the sponge through all these in-current canals. It flows out through one main opening called the osculum. So the porocytes line these holes into the sponge's body, but what creates the current? Well, what creates the current are these little cells on the inside that have this long stringy thing sticking out. These are some choanocytes. Remember, we said sponges are very closely related to choanoflagellates. And these choanocytes basically are a choanoflagellate cell. They have a flagella, and the flagella beats and kind of pushes the water towards the osculum. So this is what creates the current through the sponge's body. Part of the flagellum, the part closest to the cell, is surrounded by what's called a collar. And this collar is made up of microvilli. And the microvilli, think of them like fingers on a hand. These microvilli are kind of sticky, and they trap small food particles in the water as the water is flowing through the body. And so then these food particles are brought into the choanocyte by endocytosis. Now, the problem is, how do these cells on the outside, these pinocytes, how do they get nutrients? Well, there are some cells that can crawl through this mesohele, which is in between the layer of choanocytes on the inside of the cell and this outer epidermis made of pinocytes. So these amoebocytes pick up food nutrients from the choanocytes and transport them out to the pinocytes. So when we look at this organism, there is a little bit of cooperation. They share food. All the flagella on these choanocytes have to be with a little bit of synchronization. If they don't, the current of water that flows through the body is weaker, and weaker current means not as much water comes through, so there's not as many microorganisms to filter out of the water. Now, when we look at this, this is kind of their basic body. So think of it as being kind of like a vase. Unlike a vase, down at the very bottom, if we would continue down, there is a little section that anchors the sponge to its substrate, whatever it's living on, that board or that branch or that rock, or even just some hard dirt at the bottom of the ocean. So we want to keep that in mind. Now, when we look at sponges, they can reproduce a couple of ways. They can reproduce asexually by budding. And in budding, it's very similar to what we saw in yeast. A new sponge starts to grow out of the body of the original sponge. Cells in that area divide by mitosis, and this is what creates the new sponge. And since they divide by mitosis, unless there's some error in replicating the DNA, unless there's some mutation, if there's no errors, this will be an asexual form of reproduction. And then eventually this bud can pinch off and we get a new sponge. When it pinches off, it can drift on the current in the water for a while. So it won't necessarily land right next to the sponge that produced it. Fragmentation can occur just because the sponge isn't very specialized. If you think about the types of cells we just talked about, we only mentioned, half a dozen cells. In a person, there are 240 different types of cells. So here, if you take a sponge and you split it in half, it's quite possible that both halves of the sponge will be able to grow and form a new sponge. So in fragmentation, a piece of the original sponge breaks off, and again, it may drift on the current for a while, but eventually it settles down and it forms a new individual. And the original sponge, the cells that are left, can divide by mitosis and can replace the missing section. But sponges can also reproduce sexually. Remember, we said this was something that was a characteristic of animals in general. And when we look at this, some of the collar cells can develop into what are called spermatocytes. And spermatocytes make sperm. Some of the amoebocytes can develop into oocytes, and oocytes make eggs. The sperm are released into the water. They're just going to float on the current. But if they come in contact with another sponge of the same species, the sperm can attach and then fertilize the eggs. And then we have a organism produced by sexual reproduction, so its DNA is different because one sponge made the sperm, one sponge made the egg, and they were made by meiosis. So genetically, this new organism is different. Now, the fertilized egg develops into a multicellular ciliated larva. And when we talk about a larva, it's just a stage in development. It usually looks very different from the adult. And one of the reasons that this larva is different is because it's ciliated, which means it can use the cilia and it can swim. The outer surface of the body is covered with cilia. Although it can swim, keep in mind cilia are pretty small. Remember, they're cellular organelles, so they're not as big as a tail or a fin or a flipper. So mostly the larval stage drifts on the current. But this does allow it to move to a new area. Now, if we think about how the sponges are divided into the different classes, they're divided into classes based on the type of spicules they make and based on the complexity of their internal chambers. So the area between where the water flows in and where the water flows out. If we look at sponges that have calcium carbonate spicules, now calcium carbonate, you might have seen, calcium carbonate is one of the chemicals found in something called limestone. Although we don't use it too much more in school, calcium carbonate is what makes up chalk. So calcium carbonate is hard. And sponges that make calcium carbonate spicules actually form a large portion of what we call coral reefs. Now, coral is another type of animal that we'll talk about eventually, but part of the coral reef is not really coral, despite its name. If we have the sponges that make a skeleton formed of spongin, this is what originally were used as bath sponges, because this is a protein, this is relatively soft. And there are places, particularly in Europe and the Middle East, there may be other places, but those are the two locations that I'm familiar with, where, you know, if you go there as a tourist, you can pay somebody and they will dive underwater and bring you up a sponge. Now, the sponge you get is a living organism. So the sponge that's actually used as a bath sponge, this is produced after the living organism dies and most of the cells deteriorate. Because remember, the spicules are just protein molecules. So the protein molecules stay intact. And remember, the spongin molecules form that kind of network that we saw before. So this is where bath sponges originally came from. Today, if you go to the store and you buy a bath sponge or a sponge to clean your kitchen with, it's probably produced industrially. And a lot of sponges today are made from cellulose or some kind of chemical derivative of cellulose. If we look at the sponge body, the sponge that we were looking at the beginning was an example of an asconoid sponge. And in an asconoid sponge, there's one main chamber in the body. So we have one layer of choanocytes, these red lines on the inside. We have one layer of pinocytes, this yellow layer on the outside. All this gray is the mesohyal. If we look at a syconoid sponge, notice that you can see all these little U-shaped side channels. Each one of these is essentially like an asconoid sponge. Notice that we have all these little side openings where the water comes in. And so what we have is a bunch of asconoids attached to a bigger chamber, if you will. So this form of the body is a little more complicated. And then finally, the most complex sponge body plans that we see are the leuconoid body plan. Now notice there's always one layer of the pinocytes, the yellow on the outside. But here we have one chamber surrounded by choanocytes. We have, after the water flows through there, it flows through another chamber lined by choanocytes. So instead of having one chamber from when the water gets in to where it gets into the exit chamber, we have several chambers. So the body plan can be relatively complicated. Now, the other animals that we'll talk about are all classified in the subkingdom Eumetazoa. And keep in mind, some people don't divide the animals up into two subkingdoms, but some do. So it includes all the animals except sponges. And sometimes we don't use subkingdoms, so they're all in one group. And I think we're about out of time. We're not going to have time to finish this, so we're going to quit for today, and we'll meet again tomorrow. Are there any questions about what we've talked about with sponges or anything else with animals before I leave this screen? No? Okay. If you have any questions that you can think of about anything else we've talked about recently or anything else related to animals, you can ask me now. I should point out that, you know, a good example of how popular culture does not reflect biological reality. If you've ever seen a cartoon show called SpongeBob, sponges do not have nerve cells. So sponges don't have brains. They definitely don't have legs or arms. And not only that, you know, if you've ever watched SpongeBob, he's often out trying to catch jellyfish. There aren't any jellyfish that we know about that can electrocute their prey. So, you know, if you've been watching SpongeBob thinking you were getting a biological education, I hate to disillusion you, but it's leading you down a path to nowhere. So if there are no more questions, I'll let you go now, and I will see you tomorrow morning.
Mr. Griffith, the questions, did you send the questions already?
Which questions?
I think you said the questions are going to be due on Friday, right?
The protist?
Yes.
I think I sent them. Are they on? I'll have to check. If they're not, I'll post them, and I'll push back the date that they're due till next Wednesday. I think originally they were due on Monday, though, if I remember correctly.
Yeah, due with the essay on Monday, with the research topic on Monday.
Yeah, the bacteria questions and bacteria and virus. I'm sorry.
Okay, is it two now? Bacteria and virus.
Is it what?
Is it two? Like two papers or just one? I think it's just one, right?
Well, I think I'm going to have you submit the paper separately, just because I did ask you to do certain things with the formatting, you know, certain types of margins, certain type of font and things like that. And I think it will be easier if you submit that separately instead of just cut and paste it. So the questions actually say that, and I'll double check after we finish and see if they're posted yet. If not, I'll post them, and we'll push back the due date till Wednesday if they weren't posted already.
You said, is it bacteria and virus, or we can choose one, bacteria?
Well, the paper is about bacteria, but the other questions that you're going to turn in, some of them are about bacteria, some are about archaea, some are about viruses, some are about prions and viroids.
All right, thank you.
You're welcome.
So when is the paper due?
Next Monday, unless I didn't have that posted. If I didn't have that posted, then that will be Wednesday, because I'll have to post it now. So I always give you a week.
Okay.
Mr. Griffith,
Mm-hmm.
you said you're gonna send back my paper to me to see if I have to make any correction.
Okay. Did you send it in? Does it have your name on it?
Yes, I sent it since last week to you.
Okay, I'll double check and look for it and send it to you today.
All right, thank you.
You're welcome.